Good morning, friends and dear student. Today we'll start with the another topic from Mammalian Histology, a male reproductive system. In that, the topic is testes with reference to seminiferous tubule and histology of cells of leading and the seminiferous tubule only. Overall, these testes, male reproductive system in mammals, it consists of uh, this major portion testes, a duct system, and this prostate gland, seminal vesicles, ejaculatory duct, and urethra. So urethra is surrounded with the prostatic glands and the urethra. So apart from entire this male reproductive system, in our university syllabus, they have given more emphasis on detailed histological structure of this seminiferous tubule and a cell of leadings which lies in between that seminiferous tubule. So though we have to study only these detailed two topics, we have to cover some other points also to get the idea of entire histological of that testis. So here we have diagrammatic presentation of testes. This outer greenish color line, that is a scrotum. Testes in a mammals, they are hang outside the body in a skin pocket. That skin pocket is called as a scrotum. So you can say that testes are hang outside in a scrotum. They are two in number, packed into the same scrotum but divided with the septum. Now here in a scrotum, from outside to inside, if we start tracing the histology of these testes, what you will see is a inner layer, that is tunica vaginalis. Thereafter you will see that tunica albuginea, and in this tunica albuginea you will find there is a pocket form, empty pockets are formed which are filled with these seminiferous tubules, and we are supposed to learn the histology of this tubule. We'll learn the cross section, transverse section of this seminiferous tubule rather than learning the entire cross section of a testis. They have specifically mentioned about the cross section of seminiferous tubule. So what this you are looking at this tubule. You are in next few slides we will see detailed histology structure of this seminiferous tubule. So all these such bunch of tubules are separated and kept into this tunica albuginea in a form of a compartments which open anteriorly. They open into a network of a tubule that is called as a tubuli recti. Tubuli recti, a tube which connects seminiferous tubule with the rete testi, and rete testi is a some form of a dense matrix-like structures with a interconnecting tubule. So they form such very branch and interconnected structure of a tubules through which that secretions of seminiferous tubule slowly pass towards the outside, that is here in a afferent duct. It comes into the afferent ducts, then slowly it goes pass into the epididymis and then cauda epididymis, and finally through the ductus deferens it pass into the ejaculatory duct. So in few next slide we'll see detailed structures of this seminiferous tubule. Seminiferous tubules are, now you can understand, seminiferous tubule is what is a coiled loop structure that histologically they contain Sertoli cell and spermatogonic cells. Now this is a cross section of testes showing the presence of seminiferous tubule. Now here you need to have some imagination, like if you cut a bunch of thread into a single plane, what you will see, not a thread but a round bulb-like structure cut into the plane. So all these are the tubule cut into the section. So though they look like here a round structure, they are actually tubes cut into single plane. So these are the seminiferous tubule. Here again in this large view, that is low magnification view, you see this seminiferous tubule. All these round structures, they are seminiferous tubules. And here some part of epididymis and mediastinum has been sectioned in this slide, so it is visible here. Now, in these seminiferous cross sections, you may see here very dark spot and a pink color structure. So all that pink color structure is a cytoplasmic thing, and these dark color structures are what? Nucleus. Because of that hematoxylin stain, you get that dark color stain. Now all these small and large size violet color spot, they are what? Developing sperms. They are developing sperms. Now see here, this is a diagram you need to draw in your notebook. So I'm showing you here this diagrammatic presentation of a seminiferous tubule, cross section of seminiferous tubule. Okay. So there are many seminiferous tubule are cut into the plane. Here you can see these are all different seminiferous tubule cut into the sections, and in between the seminiferous tubule you find some kind of a cell. They have some different functions that is shown here, and the same diagrammatic presentation is drawn here. Now we'll see detailed component of this seminiferous tubule. So basically this is a round tubule like structure. Here a basement membrane is there. On that basement membrane there is a cellular layer present. Now that cellular layer here, it is called as a spermatogonic layer. Spermatogonial layer. All these spermatogonial layer is settled over the basement membrane. These spermatogonia are the precursors of that sperm. So slowly, when there goes a cellular division, that is a meiosis, slowly this, after meiosis, this spermatogonia, they develop and they form this sperm. So this is spermatogonial layer over this basement is an actual site of synthesis of sperm. Okay. Now around this seminiferous tubule, what else you see? These are the interstitial cells of leading. These yellow color bodies are interstitial cells of leading, and this structure with red spot in that is a blood vessels. To indicate blood vessels, they generally use such kind of a thing. So all these seminiferous tubule are surrounded with the blood supply. Naturally they require some blood supply and leading cells. Okay. So in seminiferous tubule you have to remember three types of cell. First is interstitial cells of leading, which lies outside the seminiferous tubule. Second one is a Sertoli cell, which lies inside the seminiferous tubules, and the main substance of seminiferous tubule is a spermatogonia. Okay, so you try to remember these three names of a cell: spermatogonic cells, Sertoli cells, and interstitial cells of leading. As what else is there? It's a basement membrane. Now, when these seminiferous tubules are observed under the scanning electron microscope, this electron micrograph shows that exact structure what we theoretically predict. Now see here, all these are the tubules break into a part. Okay, so when you open this tubule, any single tubule, you will find centrally directed lines. These lines represent for These lines represent developing spermatogonia. And in close-up view of this micrograph, you can see a presence of some different types of cell than the spermatogonial cell, that and you will find that majority of all that developed spermatogonia, they are attached with the one structure. That structure is what it is, Sertoli cell. Now see, this is a magnified view. Now all these are the seminiferous. This is some seminiferous tubule and magnified view of seminiferous tubule. Now here these all are spermatogonia in different phase of development, and centrally all you see this tail or filament structure. These are the tails of a sperm, fully developed sperm. Now see here, you can see the fully developed sperm is there. These sperms are attached to some kind of a structure here, and wherever they are attached, that is a Sertoli cell. Here you may not see it very clearly, but in next few slides you will see. Now see here, they have shown how that sperm synthesis take place. That is seminiferous epithelium they have shown, along with the, so this is a basement membrane. Over the basement membrane there is a layer of this spermatogonia. Okay, and in between this spermatogonia, all they are embedded into the matrix here, which is full of a blood supply. So you can separate this layer of the seminiferous tubule into two parts as a basal component and adluminal component, towards the lumen, towards the cavity component and towards the basement component. Now Sertoli cell, these Sertoli cells are there, and in all these spermatogonia are embedded in Sertoli cell. You carefully observe, if you keenly observe this slide, you will find that this is a nucleus of Sertoli cell, and all these reddish color structure with red color dot, these are all cytoplasm of this Sertoli cell. Now basic functions of this Sertoli cell is to provide nourishment to developing spermatogonia. Are you getting the point? All these developing spermatogonia require some nourishment, and that they cannot derive from the blood. So there is a Sertoli cell. Through the cytoplasm of Sertoli cell, the nourishment is provided to these developing primary spermatocyte, secondary spermatocyte, and spermatids. So all these mature sperms then remain attached to the matrix here, and the matrix is of a Sertoli cell. Now in the cross section, enlarged cross section of the seminiferous epithelial cell, seminiferous tubule, you will clearly see that seminiferous epithelium and the labeling. So this central portion where you will find all the tentacles or all the tail portions of sperm is there, and these sperms are attached to some body here. Now here you can see they are directing to some portion here. This portion is a Sertoli cell. They are not a single column like structure, but they are spread and matrix like structures of a Sertoli cell. And through the by just like touching through the Sertoli cell cytoplasm, there develops this spermatogonia. So everywhere you will find these spermatogonia are in a different stages of meiosis, cell division stages. So here it is a spermatocyte. Now see here they are developing stages of spermatocytes, and this is a spermatogonial layer. This is early spermatocytes. Here again you can see the early spermatocyte, late spermatocytes, and completely fully developed formed sperm and the lumen. So through this lumen only that sperm travel towards the ejaculatory duct. Now here in this photograph again the Sertoli cells and spermatogonia are very enlargedly shown. So first we'll see here Sertoli cell structure. Now you can say clearly distinctly observe the different pattern of this color. So here these are the spermatocytes, and this pinkish color with a round nucleus, these are the nucleus of Sertoli cell. And all cytoplasm of Sertoli cell is spread around this area through which these spermatocytes are developing. And between the spermato between the two three seminiferous tubule, you will find there are different kinds of cell type, that is the leading cells you will see there. Now, what is the function of these leading cells? This function of leading cell is to secrete testosterone, a male sex hormone. In puberty, in response to that gonadotropic stimulation from the pituitary, these leading cells get stimulated and they start synthesizing and secreting androgens. They also secrete primary testosterone, and these leading cells number remain constant throughout the life of a male mammals. They have large spherical nucleus and that contain a rim of heterochromatin which helps them to identify during staining. So in this electromicrograph, you can see this spermatogonia and a Sertoli cell. Okay. So this all whitish color portion is a Sertoli cell cytoplasm, and this dark color portion, these are the spermatogonia.